Hey guys, sorry I'm looking a little rough today. I was up all night with a sick child who is coincidentally watching number blocks on the TV right now. So if you hear the TV in the background and you're thinking, sounds like kid shows, it's because it is. So what I'm going to do is I'm going to go through question by question on this study guide, and we will go over that. We'll give you the answer to all of them. And if you have any questions, shoot me an email and we'll go over it. I'm going to make a separate video going over the review for the bonus. All right, so first things first, you need to print out your study guide. So if you have not done that yet, you need to do that and then come back to this video. Okay. Once you get that done, come back to the video and we're going to fill it all in, okay? So number 1, who is the scientist who first theorized heredity using pea plants? That was Gregor Mendel. When you hear genetics, it's usually referred to as Mendelian genetics. So Gregor Mendel is the person who theorized heredity using pea plants. You can give me Mendel, you can give me Gregor Mendel. I don't care which. The next question, the alternative version of a gene is called an allele.
Mom, no.
Hold on, let me change the TV for him. You want animals?
No, I want toy trick shot.
Sorry, guys.
You have to say toy trick.
No, you just have to pick one because I'm making a video for my class right now, okay?
I just want toy trick shot.
Dude Perfect toy trick shots. Sorry. That one?
Yeah.
Okay. Sorry about that hiatus. All right. Number 2 we did is an allele. It's the alternative version of a gene. All right. Heterozygous genotypes will be represented by a big letter and a small letter. So big B, little b. And it will express the dominant phenotype, because if there's ever a big B, it'll always be the dominant, right? Those recessive traits get masked by the dominant ones if they're ever there. Okay? Number 4, complete the Punnett square. You've got big B in the top, big B in the top left, big B, little b in the top right, big B in the bottom left, and big B, little b in the bottom right. So that gives you a genotypic ratio. So when you do genotypic ratios, remember the ratio goes homozygous dominant, heterozygous, homozygous recessive. So the first number is going to be the number of homozygous dominants. The middle is going to be the number of heterozygous. And the one on the right is going to be the number of homozygous recessive. So here you've got a 2-to-0 genotypic ratio. Next, for your phenotypic ratio, it's always going to be the number of dominants and then the number of recessives. So you've got 4-0 on this one because all of these offspring are going to show the dominant trait. Sorry. Woohoo! Okay. Phenotype is the term used to describe the physical characteristics of an individual. Genotype is the term used to describe the combination of alleles that represent a trait found in our DNA. So, phenotype is physical, genotype is the genetic coding that make up. So, all right, the next one we didn't really go over in class, but it should be pretty self-explanatory. This law states that allele pairs.
I want to stay out of school 2 days.
We'll see. States that allele pairs segregate or separate from each other during gamete production. So that law is called the law of segregation. Pretty self-explanatory. Law of segregation states that those allele pairs segregate when the gamete is formed. All right. Next,
Yeah, be going to feel like.
you've got the Punnett square, big R, little r, little r, little r, big R, little r, little r, little r. So you've got big R has brown, you have brown hair. Little r is red hair. All right. How many of the offspring will show brown hair? So you've got 2 that have the dominant trait that are going to show the brown hair. And you've got 2 that are homozygous recessive, so they're going to show the recessive trait. It has to be homozygous recessive in order for the recessive trait to show up.
Yeah.
So you've got 2 that are going to have red hair and 2 that are going to have brown hair. But with that being said, just because somebody has brown hair doesn't mean that they don't carry the trait for the red hair. So those 2 that are showing brown hair are heterozygous because they have big R, little r. So all 4 of them will pass on the trait for red hair. That's just how genes work. So think of it this way. So you see in this video, my eyes are brown, right? My husband has blue eyes. So you would think, okay, brown eyes are dominant, blue eyes are recessive. So my children would both, if I'm homozygous dominant, then both of my children would have brown eyes. But both of my children have blue eyes. So that means even though I show the dominant trait, I carry the recessive trait. Yes, you are my child. So both of them have blue eyes. So that means that even though I show brown, I carry the recessive. So that recessive got passed up to my kids. Where you going, buddy?
Can you turn your track into a new track?
Okay. He's going to go build Hot Wheels tracks now. So, sorry. This is the best I can do today. All right. So, next we've got the Hardy-Weinberg principle and the 5 assumptions that it must have in order for it to work. Populations are not going to ever be a Hardy-Weinberg population outside of the ideal.
Mom, look.
Good job, buddy. So, the assumptions are that there has to be random mating, has to have a large population, no genetic mutations, no natural selection, and no gene flow. Hey. I'm making a video for my class. Can you just turn your volume down just a smidge? Thank you. All right, so no natural selection, no gene flow, random mating, large population, and no mutations, okay? I'll give you a second to write that down. All right, so the next one, the Hardy-Weinberg equations. You've got P plus Q equals 1. And then you've got P squared plus 2PQ
Mom, pause it. I need that.
plus Q squared equals 1. So P plus Q equals 1. P squared plus 2PQ plus Q squared equals 1. So sorry. Okay. So you're thinking, okay, why are there 2 different equations? That first equation, P plus Q equals 1, is to find the frequency for one allele. The P squared plus 2PQ plus Q squared tells you the frequency of homozygous dominant versus heterozygous versus homozygous recessive. So that's going to tell you the combination, the frequency of the combination of alleles. So for 15, the variable for one dominant allele, that's P. So one recessive allele is Q. So if following that logic, if one dominant allele is P, then 2 dominant alleles would be P squared. Sir. One dominant allele is P, 2 dominant alleles is P squared. So again, with recessive, if one recessive allele is Q, 2 recessive alleles is going to be Q squared. But then the variable for the heterozygous, or one of each, is going to be 2PQ. All right? Hey, Hey, turn your volume down a little, okay?
Okay.
Thank you, sir. I'm so glad you're feeling better.
Put this on the other couch.
I don't mind. Yes, you can do that. All right, number 20. The two scientists who are credited with discovering DNA structure. Hey, be careful with that because I have something drawn on that for this video, okay? Just put it in the floor. Thanks, Nathan.
So you could see it?
Yeah, I'm going to put that up
They're
here.
all exposed.
Yeah, can you put the expo markers down there with that too? Thank you.
I wish I do.
Perfect. Okay. The two scientists who are credited with discovering the DNA structure, Watson and Crick. And they discovered the double helix, the twist in it. Watson and Crick. All right. So the next one. Recessive alleles may be masked by dominant alleles, but they can be passed to the next generation. Like we just talked about with that Punnett square. Hey, that's a little loud. Where just because I have brown eyes doesn't mean I don't carry the the allele to be recessive, because both of my kids show recessive. So, okay. Big R represents homozygous dominant. Don't just tell me homozygous and don't just tell me dominant. I need homozygous dominant with that question. Because if you just tell me homozygous, there's another form of homozygous. It's homozygous recessive, and it's little r. I will count it wrong. Okay? Big R is homozygous dominant. Big R, little r, so one of each, that's heterozygous. Heterozygous. So think of it as, hetero is different, homo is same. So heterozygous, one of each allele. Okay? Darwin is the scientist most closely associated with natural selection. Charles Darwin, when you think of evolution and when you think of natural selection, he's usually the one that pops right into your head. Okay, 25. The fitness of an organism includes any features that lead to successful reproduction in a certain environment. Remember, we talked about this. You want to have good fitness in the environment. You want to set yourself up for success, so you study for your test. You want to be successful on the football field, so you work out. You want to be successful on the track, so you run. You want to be successful on the court, so you shoot free throws whenever you have free time. That fitness, you get your body into the best physical shape that it can be in for your sport or whatever you are doing. If you're more of a scholar than an athlete, you study for your stuff, so you've exercised your brain in order to know that this is the answer. So the fitness of the organism is any feature that leads to successful reproduction in a certain environment. So it gets itself ready to survive and thrive in the environment it's in. Okay? 26, survival of the fittest is the phrase most often used to refer to natural selection. All right, survival of the fittest, right? We know that. Blank is the total number of individuals an ecosystem can contain based on available resources. I know I talked about this because I always use the drink analogy of there's only so much plastic in a 16-ounce water bottle, but if somebody's got a 40-ounce Stanley, they're going to get more water because they got more metal than you got plastic. The carrying capacity of those set that ecosystem up to hold that much stuff. So the carrying capacity is the total number of individuals an ecosystem can contain based on available resources. All right. 28, rapid evolution in predator and prey relationships happen because pressure for prey to avoid and pressure for predators to be able to get to their prey. So rapid evolution happens fast. All right. 29. The survival of an organism means its genes survive, not necessarily the organism itself. So those genes are what they want to pass on. Those genes need to be what survive, okay? Nathan. All right, number 30. Charles Darwin observed finches to observe the difference in beak sizes. So he studied finches. All right, biology is the scientific study of life or living things. Cells are the basis of all living things. All right, we're about to get into histology, and that is a big, hairy deal on this test, okay? You really, 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 really need to study the Lab 7 histology PowerPoint. Those pictures of the muscles, of the tissues and stuff, the pictures of the different tissue types and shapes and sizes. That is a huge deal on this test. So just go through on that histology slideshow, make yourself some flashcards with those pictures or however you need to do it. That is how you need to study those pictures, okay? Because those pictures are going to be on this test. I'm just going to go ahead and tell you. Hey, if you'll go get it, I'll pour it in there. It's in the kitchen. All right. So, cells are the basis of all living things. Cells that get grouped together based on similar structure and function form tissues. So, cells are the building block of life. Cells that do the same thing, have the same structure function, they form tissues. Tissues form organs form organ systems. Organ systems form organisms. So, cells make tissues. Tissues make organs. Organs make organ systems. Organ systems make organisms. Okay?
Mommy,
Yes, honey.
can I use the Powerade?
Yeah, boo, bring it here and I'll pour it in there for you.
I will already going to pour it.
He's going to pour it himself. So here's to hoping that I don't have a Powerade spill in my kitchen floor in about 5 seconds. All right. So next we're going to look at number 34, the study of tissues. It is called histology. Okay. And tissues provide numerous functions necessary to maintaining biological life. Yes, baby, if it fits. Okay. Histology, the study of tissues. Okay. All right, number 35, we're looking at the 4 basic types of tissues in the human body. You have epithelial tissue, connective tissue, muscle tissue, and nervous tissue, okay? Epithelial, connective, muscle, nervous. Those are the 4 basic tissue types in your body, okay? So next we're going to look at the function of each of these, all right? So the function of epithelial tissue is to line and cover, okay? Line and cover, epithelial, line and cover.
Kirby wins to all. It's all.
All right, go sit down.
Kirby wins it all.
Kobe wins it all. Dude, perfect. All right. The function of epithelial tissue is to line and cover surfaces. All right. The function of connective tissue is to protect, support, and bind together. Okay? So it protects things, it supports it to keep it built up, and it binds it together. All right? The function of muscular tissue is to move things, okay? That makes sense, right? When you think of muscles, you think of movement. All right? Nervous tissue is to receive. I can't talk. I've been up all night. The function of nervous tissue is to receive stimulus and conduct impulses, okay? Your brain. Think about your brain, all right? So next, number 40, epithelial tissue is classified according to shape and arrangement, okay? Shape and arrangement. There are 2 sub-classifications of classifying these things, okay? The shape classifications for epithelial tissue are squamous, which is kind of squished, cuboidal, which looks like a cube, and columnar, which looks like a column. So they're pretty self-explanatory, okay? The arrangement classifications are, they're simple, so just kind of one layer. Stratified, which has got layers. Pseudo-stratified, which looks layered but kind of isn't. And transitional. Transitional is kind of special in its own way because it's really for like stretching and dissension. So that's your bladder, pretty much. All right, the classification for connective tissues, number 42. Connective tissues, you have loose connective tissue, dense connective tissue, cartilage, and blood and bone. Those kind of get lumped together, blood and bone too, but they are connective tissues. So think about what the function of connective tissue it supports, it protects, and it binds together, right? So loose, dense cartilage, blood and bone, all right? The types of connective tissue, all right? Areolar, 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 adipose, which is just fat, and reticular. Those are the types of loose connective tissue. All right? The types of dense connective tissue. These make a little more sense. Regular and irregular. Okay? Types of muscular tissue. You've got skeletal, which is what you think of when you think somebody's got muscles. Those are your skeletal muscles. You've got your smooth muscles, which are like your GI tract that moves things along. And you've got cardiac. Cardiac is its own type. Okay? 46, speaking of cardiac. Cardiac muscle is the muscle tissue that is responsible for beating, propelling, and circulating blood throughout the body. Okay? Skeletal muscle is the type of muscular tissue that provides movement to body parts. All right. Number 47 on the start of page 4. Smooth muscle is the muscle that propels substances in like the digestive tract and in your blood vessels. Okay. It's got to be smooth because that stuff's got to move easy. Okay. 48, I want you to know what nervous tissue does, and there was no way for me to really make that a question instead of just saying know it. So muscle tissue, think about it in a situation where you are feeling something. Like, say you close your eyes, like in the Dude Perfect video that we just watched, they stick their hands into a box and try to identify the animal. So they can't see it, but they've got to use their nervous tissue to figure it out. So they have sensory receptors to monitor changes inside and outside the body. So if changes happen outside, changes happen inside, your muscular tissue can regulate your body, okay? So it processes sensory input. So if I'm sticking my hand blindly into a box and there's a snake in the bottom, which there was, I feel it and my brain processes what it feels like. And then it maintains homeostasis by acting as a regulatory control center. So your brain, okay? Your brain keeps you level in moments, and then it affects an appropriate response to stimulus. So I've got the stimulus of I'm touching a snake. My reaction is going to probably be breaking my hand, getting it out of the box fast enough, and yeeting it across the room. Like I don't do snakes, not even a little bit. So that's just the best way to, or like, you know, when you put your hand on a hot eye, your brain says, hey, stupid, that's hot, move your hand. And so you move your hand. So the response to stimulus, you move your hand. The response to stimulus, you pet the snake if that's your thing. You do you, boo. Pet the snake if that's what you want to do. Couldn't be me. All right, number 49. This is going to be on slide 8 on the histology slideshow. Simple squamous epithelial tissue functions to allow passage of materials through diffusion and filtration in sites where protection is not important, and it secretes lubricant. So simple squamous, it's simple, it's one layered, and it's squamish, so it's squished. So it's not going to do much to protect you, It's not going to do much to pad or insulate. So the sites where protection is not as important, you're going to get the simple squamous. And it allows things to pass through it. Because it is simple and squished, things can pass through it a lot easier than it could, say, a stratified cuboidal or whatever. It's going to be easier to pass through a simple squamous layer of tissue than it would be something thicker. Okay. Number 50 is also on slide 8. Simple cuboidal epithelial tissue functions to secrete and absorb. Okay. Number 51, and I want you to know these pictures too. These pictures on these slides are why I'm telling you these slide numbers. These are the ones. These are they. Please study those pictures, okay? All right. 51. Simple columnar epithelial tissue functions to absorb and secrete mucus and enzymes, and it has cilia to propel substances. So those little hair-like projections, that cilia, it's going to move things, all right? That's the columnar, simple columnar, so it's going to be a little longer. That's also on slide 8, okay? On slide 9, you'll see that stratified squamous epithelial tissue protects underlying tissues and areas subject to abrasion, okay? So things that are going to get scraped and whatnot, you're going to have that stratified squamous tissue surrounding more vital things, okay? Because it's harder to get through things that are layered, right? It's easier to cut a slice of cheese than it is to cut an onion. Onions have layers, yeah, get it? There's my Shrek reference for the day. That's on slide 9. All right, 53. Slide 10 is going to show you transitional epithelium and what it looks like, and that it functions to stretch easily and permit dissension. So again, I said that's pretty much the bladder. Fun fact, when you feel like you can't hold it anymore and you're going to have an accident, your bladder is only about half full. Just thought I'd pass that along. All right. The next one, loose areolar connective tissue, is the packing material in the body. Not the fat, the packing material. So it's more of that fascia layer, that protective layer under the skin before you get into the organ systems. It attaches to skin, to underlying body parts. So it's that layer that connects your skin to the rest of your body, so your skin's not just willy-nilly all over the place. All right. Loose adipose tissue. So that's fat, remember? So what does fat do? It fuels, it insulates, and it cushions. Okay? Your body uses fat as fuel, your body uses fat as insulation, and your body uses fat as a cushion. Okay? Number 56. Hyaline, Hyaline, hyaline cartilage. It supports and reinforces. Okay? You've got hyaline cartilage and you've got elastic cartilage. So the hyaline cartilage is a little more structured. So it supports, it's for support and for reinforcement. Okay? Elastic cartilage you use to maintain shape and structure while providing flexible support. Okay? Elastic is more flexible. Okay? All right, DNA. What does that stand for? Deoxyribonucleic acid. DEOXYRIBONUCLEIC. Deoxyribonucleic acid. DNA. All right? These enzymes are the enzymes that cut DNA at certain sequences. Those are the restriction enzymes, okay? Restriction enzymes cut DNA at certain sequences. And DNA is double-stranded. All right? Number 61. Adenine, thymine, cytosine, and guanine. Those are the 4 nitrogenous bases that link DNA together. Okay, they are nitrogenous bases. So that means what element is found in all 4 of those bases? Nitrogen. All right? Nitrogenous base, nitrogen. All right? So if you watch the video like you were supposed to last week, you'll have seen my DNA drawing. The sugar phosphate is the type of backbone that makes up DNA strands. Okay? I've got it drawn out again, and we'll go over that in a second. That's what Nathan had to move off the couch. All right? DNA strands are anti-parallel and complementary, right? So that 3' and 5' carbon are always, they're anti-parallel, so it's not going to be 5' and 5' on the same across from each other. It's going to be 5' and 3'. And down here, it's going to be, I said 5 and 3. Down here, it's going to be 5 and 3. So it's going to be if 5's up here, 3's down here. 5, 3, 5, 3. So there's going to be like that. And then it's going to twist in real life. It's going to go for it. All right. Gel electrophoresis is the lab technique used to separate DNA, RNA, and protein fragments based on their size. All right. Gel electrophoresis of DNA causes DNA to travel towards the positive end of the gel because DNA on number 69 is going to typically be negative, okay? DNA is negatively charged, so it's going to run to the positive, right? So not to skip ones. 67, that's telling you which bases bind to which ones. They don't just bind willy-nilly. Remember how I said DNA is anti-parallel and complementary? I went over the anti-parallel with the 5 and the 3. Complementary means that those bases will always pair up the exact same way. So adenine will always pair with thymine. Cytosine will always pair with guanine in DNA. Okay? It doesn't matter which side of the backbone they're on. If T's over here, A is going to be over here. If T's over here, A is going to be right here. If C's over here, G is going to be right here. If C's right here, G is going to be right here. All right? They will always complementarily pair that way. Well, let's finish this one and let me get done with this video, okay? I'm almost finished, buddy. All right? So C binds to G, T binds to A. I don't care how you remember it. I don't care. At is a word. C and G look alike. However you need to remember it. Just know C and G are always together. A and T are always together, okay? Okay. The bonds that bind the bases together that hold one side to the other, those are hydrogen bonds between the C's and the G's and the A's and the T's. Those bases, those complementary bases, form hydrogen bonds to hold each other together, okay? All right, DNA is negative. We just talked about that. All right, the shorter the strand of DNA, the faster it will move through the gel in gel electrophoresis because it doesn't have as much of a chain behind it to take with it, okay? Almost done, guys. You'll need to know how to look at a gel electrophoresis handout. And if I were to say, here is the sample we got from the crime scene. Here's suspect 1, 2, 3, and 4. Which one matches up the best? You should be able to look at that and tell, okay? These bonds are the bonds that involve oxygen and hold the sugar phosphate backbone together. Those are phosphodiester bonds. Remember, we talked about that in the video. It's the bond between... Here's where the drawing comes in handy because the next question is let's draw DNA together, right? So, let me get... Oh, I'm sorry. All right, here's my drawing. I'm so thankful that my kids love to draw on whiteboards because we have a big one. All right. All right. Let the Christmas tree not be reflecting on it.
Mom, watch.
Good job, buddy. All right. So, remember, here's the 5'. So, across from 5' is going to be 3'. On the bottom of 3' is going to be 5'. And across from 5' is going to be 3'. So, there's your anti-parallelness, okay? So, here's your hydrogen bond holding these two bases together. right? You see that? All right. So the building blocks of DNA are nucleic acids. We talked about that. So the nucleic acid consists of a sugar, a phosphate, and a nitrogenous base. And those three are a nucleic acid. So the bond that holds one building block to the next building block is here. This purple right here. And that purple bond is called a phosphodiester bond, okay? Phosphodiester. And those are the bonds that hold together the backbone of DNA, all right? So, with that being said, let's look at this drawing. I'm trying to angle it to where the Christmas tree doesn't reflect in it. Sorry, guys. Yeah, we've had our Christmas tree up a hot second because we have 2 small children that love it. So, okay. This is, when I say let's draw DNA together, that's the next question, right? I want this ladder, and I want it drawn at least like this with at least 3 nitrogenous base pairs, okay? On your test, I'm gonna be specific about what I want you to label. All right. I want you to label the sugar phosphate backbone, which is the whole thing. All right. You see it labeled over there? The bracket that labels the whole thing. I'm gonna want you to label the phosphodiester bond. I don't care if you color coded on the test, as long as you label it correctly. I don't care. All right. I want 3 base pairs on the ladder, and I want them paired up correctly. I want you to know where the hydrogen bond is. I want you to see that you know that it's the 5' and 3'. I want to see that you know how those line up. And then I want you to twist it. All right? I know that sounds weird, but I'm going to want you to draw it in real life. Because DNA in real life doesn't look like this. Remember we talked earlier about Watson and Crick discovering the... Oh, sorry, I feel like I'm really close. Discovering the form of DNA, how it's a double helix and how a helix is twisted. So twist it, okay? And I want those base pairs on there too. So draw it just like that and label it just like that if you want to, or if you want to color code them, just tell me which one's which. I just want to see that you know that how DNA looks like here and here. So here's what, this is a good way to say this because I had some confusion on Monday. This is what DNA looks like in real life, okay? It's twisted, and that's how the base pairs pair up, right? So what we've done here is we've zoomed in on part of it, and we've got this to show you the pieces and to be able to see the building blocks of what makes up that. We've zoomed in, and we've stretched it out to where you can see what it is, okay? Good with that? All right. I'm going to keep this on here for a second so that y'all can take a picture, take a screenshot. I don't care.
Leader.
All right. So the next question on the study guide. I feel like the next questions are mostly about, those are the bonus questions from last year. Y'all are not getting those questions. Y'all are doing the problems for bonus. So there will be no bonus questions on this test. All of your 15 points of bonus are coming from those problems that I'm going to go over in a separate video, okay? So I'll tell you that Darwin wrote on the origin of species. Skip the next 4. 120/80 is the blood pressure considered the cutoff before it gets too high. All right? So we talked about DNA being double-stranded. RNA is single-stranded. It also uses a different base instead of thymine. It uses uracil, where it just takes the place of the T, and the uracil binds with the adenine instead of the thymine. But I'm not getting into that. That's not something that we have to worry about today or on this test. So that's just a fun fact for you. Hopefully that might help you out in lecture. All right. That's all you need to know for your study guide. Watch this video however many times it takes. Fill out your study guide. Study your study guide. You'll make 100 on the test, plus some if you bring in your problems. Bring in your bonus problems. I will not accept them on an email. I will not accept them in person before the test. You turn in your bonus problems with your test the day of your test. Your test is on the lab day during lab time. the week after Thanksgiving. There's not a special schedule for lab finals. You just show up for lab the week after Thanksgiving and we take a test. All right. Again, if you have any questions, comments, concerns, doubts, fears, anxieties, shoot me an email. My email is cchandler@shorter.edu. It's on my syllabus, which is the very first thing listed on Canvas. But my legal first name is not the name I go by, and my legal first name is what they used for my email address. So it's cchandler@shorter.edu. Your professors, our email addresses are all the same format. So it's the first initial of our first name, our last name, and it's @shorter.edu. We don't do the @hawks.shorter.edu. That's just for students. We're just @shorter.edu. All right. Remember, shoot me your questions, and I will see y'all the week after Thanksgiving. Have a very happy Thanksgiving. I'll try to answer any questions as much as I can over Thanksgiving break. Just because I'm not technically on working doesn't mean you can't shoot me an email and I won't at least see it and try to figure it out. All right. Have a wonderful rest of your week and a happy Thanksgiving, and I'll see y'all the week after. Bye, guys.